Hello, General Biology, and welcome to the fourth in the screencast series of Chapter 7, Sections 7.3 and 7.4. And in this screencast, we're going to talk about osmosis. And osmosis is water. And notice I have the water done in blue through a selectively permeable membrane. So this is a form of diffusion, which means it's a form of passive transport. It doesn't require any energy, and it's going to be the movement of water. And this is important because I think this can get a little tricky because we're going to talk about concentrations of other solutes that are present. And when we think about those other solutes, we may have more of a solute, but that means we're going to have less water. And it's the water that's going to be moving through the membrane. Or we could have less of a solute and more water, and we're not going to worry about the solute so much as where we have the more water to move across the cell membrane. So I Googled this image, and most of my images that I use are Googled. They're not necessarily from your textbook. And here in osmosis, we have a selectively permeable membrane. What did we just describe as a selectively permeable membrane that's in cells? Did you say cell membrane? Oh, you're just too good. All right, so cell membrane, it's selectively permeable, or even the cell wall is selectively permeable if you said that as well. But here we have a selectively permeable membrane. We can see the openings or pores. And the larger objects that we have are a concentration of something. It could be sugar, it could be some other type of nutrient that needs to come in. And then the blue little dots that we have here is the water. So let's take a look at this picture. We have a high concentration of the solute on this side of our selectively permeable membrane. We have a low concentration of the solute here. So what that means to us is if we have a low concentration of the solute here, we're going to have more water molecules. If we have more of the concentration of the solute here, we're going to have less water molecules. And visually we can see this. If we count it, there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 water molecules here. And there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19. Well, I think you get the point. There's more over here on this side. So when we talk about the pathway of them moving, how are they going to move? It's going to be from this high concentration across the membrane through the pores to the area of low concentration. Notice that the solutes, they can't fit through, but the water molecules can. So the water molecules will pass through this membrane through the process of osmosis. So why is that important? Well, let's take a look at this. It creates osmotic conditions in the cell. So here's our selectively permeable membrane again. And when we look at this, here is our concentration. So we've got a low concentration of solutes here, a high concentrations of solutes there. So where do I have more water, on side A or side B? Hopefully you're saying side A. So if I have more water on side A, it's going to pass through the selectively permeable membrane and it's going to go into side B. Look what happens to the water level. The water level drops here and the water level rises here. So that it might not look equal, but because of the concentration of solutes here, the amount of water will be equal on both sides. And what this does is it creates something known as osmotic pressure. And that pressure is going to help provide shape and structure to the cell. Okay? You need to know that cells are constantly surrounded by water and other solutes. Remember, we talked about the cytoplasm being a suspension. So that's where this is important in knowing that there's solutes in there. And the concentration of those solutes and the amount of water will impact the direction of flow of that water into and out of the cell across that selectively permeable or semi-permeable membrane. Okay, so we're not done. We don't end there because this is science, this is biology, and there's so much to add. So with osmosis and cells, there's 3 different kinds of osmotic pressures or osmotic conditions that can be created. And they are a hypotonic condition, an isotonic condition, and a hypertonic condition. So I liked this illustration. We're going to start with the hypotonic solution. And in a hypo, when we think of hypo, think of a hypodermic needle, okay? It's going to go into, when you get a needle, it's going to go into your cells. Tonic means solution. So this is a hypotonic solution where the concentration of water is going to be more on the outside of the cell than it's going to be on the inside of the cell. So there's more water here and there's less water there. So the movement of water is going to go into the cells. So let's think of a balloon. If we kept putting air into the balloon, what's going to happen to the balloon? Yeah, it's going to get bigger and eventually it's going to burst. Exactly. So cells that are in hypotonic solutions are going to swell and burst. Plant cells, the plant cell wall might prevent it from bursting and you might just see it swelling and bulging, but in animal cells it's going to swell and burst. So these are pictures of red blood cells, and this one is getting too much water going in and it burst. It broke open. Now an isotonic solution, iso means the same. So that means you have an equal amount of water in the cell as you do outside of the cell. So it's equal on both sides. So that means that there's not going to be any overall net movement where you're not going to see a big change because the flow is going to be equal. You're going to get the same amount coming in as the same amount as going out. And this would be a cell under optimal conditions. This is what we want, under the best conditions in order for it to be able to survive. But we know that that doesn't always happen. The cell's going to be really stable, it's going to be able to function, the osmotic pressure is going to be balanced, and it's going to be a happy cell. Another type of osmotic solution is a hypertonic solution. And I lost my arrow, there it is. And in a hypertonic solution, think of a hyperactive child. They're going to get all of their energy out. So this is where the water is going to leave the cell. You've got a greater concentration of water inside of the cell, less of a concentration of water outside of the cell. So the cell is going to lose the water and it's going to shrivel up and shrink. And that's a hypertonic solution. It flows out. And we can see that these blood cells are shriveling and shrinking, like letting the air out of the balloon. Now in plant cells, this is important because it creates a condition in plant cells called plasmolysis. So we've got to remember that plant cells have a cell wall that's a rigid cell wall. And that cell wall is going to remain in place. But all of the cell contents are going to shrink and shrivel up. And that's what happens with plasmolysis. The membrane maintains its shape, but everything else shrivels up. So again, when does plasmolysis happen? in a hypertonic solution, and I think plasmolysis, PLA, is the start of plants, PLA, plants, okay? In a hypotonic solution, it's going to cause something called turgid. This is where we get our turgor pressure from, and hopefully you saw that little connection there. All right, I believe that this video is a little bit longer than what we have left, so we're going to end here, and I will start this video in the next screencast.